Hello, and welcome back to another episode of Teach Me in 10. Here, we invite scientists to describe their research in less than 10 minutes. I'm Bree Foster, a science writer at Technology Networks, and today we have a fantastic guest with us, Leah Cronan, a product specialist at Promega who manages the molecular cloning products line. Today, she will discuss the fascinating world of RNA therapeutics, explaining how messenger RNA molecules are produced, the importance of cell-free RNA synthesis, and key considerations when scaling up RNA production for therapeutic use. Thank you so much for taking the time to join me on Teach Me in 10 today. I've got a bunch of questions I'm excited to dive into, so let's jump right in. First off, what does a typical mRNA molecule in an RNA therapeutic actually look like?
The RNA in an RNA therapeutic is a single-stranded messenger RNA molecule that is engineered to carry genetic information that directs the synthesis of a specific protein. The important structural components of the mRNA molecule include 5' cap structure, which is a modified guanine nucleotide that's added to the 5' end of the mRNA. This piece is crucial to mRNA stability, efficient translation, and protection from degradation by exonucleases. The RNA molecule also has a 5' untranslated region, which is just upstream of the protein's coding sequence. This region aids in the regulation of translation and influences the efficiency of protein synthesis. There's also a coding sequence, which is part of the mRNA and is then translated into a protein. It contains the open reading frame, or the ORF, that encodes the target protein. The 3' untranslated region, or the UTR, is another region which is located downstream of the coding sequence. The 3' UTR is important for mRNA stability and the regulation of translation. There's also the poly A tail, which is a stretch of adenine nucleotides that are added to the 3' end of the mRNA. It is responsible for protecting the mRNA from degradation, which aids in the initiation of translation.
Thank you so much. Why is cell-free production the preferred method for making RNA in therapeutics?
Cell-free production of mRNA is also known as in vitro transcription, or IVT. IVT is the preferred method for producing RNA for RNA therapeutics because IVT is scalable, it's fast, and it's flexible. So it's a method that allows for consistent large-scale RNA production. And going into a bit more detail, cell-free systems are scalable, right? So that means they can easily be scaled up to produce large quantities of RNA, which ensures a sufficient supply of RNA for therapeutic applications, including large-scale vaccination campaigns and clinical trials. Production using cell-free systems is consistent because it allows for precise control over the synthesis process, leading to high batch-to-batch consistency. And this is crucial for ensuring the safety and efficacy of RNA therapeutics. Cell-free systems also minimize the risk of contamination with host cell proteins, DNA, and endotoxins, which can be problematic in cell-based production systems. And cell-free systems are fast, so they can produce RNA much faster than cell-based systems because cell-free systems bypass the need for complex cell culture steps, including cell line selection and condition optimization. And cell-free systems are flexible. They can easily be customized to produce various RNA molecules just by changing the DNA template.
Okay, well, that sounds really interesting. So, when you're doing in vitro transcription, what are the key components of the reaction, and what role does each of them play?
So, an IVT reaction contains a few necessary raw materials to synthesize the RNA molecules. The first reaction ingredient is the DNA template, which serves as the blueprint for the RNA synthesis. The template contains a promoter sequence that is recognized by the RNA polymerase. RNA polymerase is an enzyme that catalyzes RNA synthesis by recognizing and binding to the promoter sequence in the DNA template and incorporating ribonucleotide triphosphates, or RNTPs, that are complementary to the DNA template strand. RNA polymerase then continues down the DNA template path, synthesizing the RNA molecule by catalyzing the formation of phosphodiester bonds between RNTPs in the 5' to 3' direction. The RNA polymerase will terminate RNA synthesis when it reaches a transcription termination sequence in the DNA template. So for the RNA polymerase to function efficiently, it requires an optimal reaction environment. And this reaction environment will contain the ribonucleotides, the RNTPs, that serve as the building blocks for RNA. It will also have magnesium chloride, which is a required cofactor for RNA polymerase activity, and the reaction buffer, and that helps maintain enzyme stability and maintain salt and pH conditions. It can be beneficial for an IVT reaction to contain two additional enzymes, inorganic pyrophosphatase and an RNase inhibitor. During the RNA synthesis process, when the RNA polymerase catalyzes formation of the phosphodiester bonds between the ribonucleotides, RNA polymerase releases inorganic pyrophosphate as a byproduct. And accumulation of this inorganic pyrophosphate byproduct can actually inhibit the RNA polymerase activity. So inorganic pyrophosphatase is an enzyme that helps to maintain the equilibrium of the in vitro transcription reaction by hydrolyzing this inorganic pyrophosphate into two inorganic phosphate molecules. So it prevents the inhibition of RNA polymerase and allowing it to continue with RNA synthesis. And the RNase inhibitor is an enzyme that can prevent RNA degradation and increase RNA yield by inhibiting ribonucleases that can be introduced into the reaction by contamination.
What methods are used to measure the success or failure of a synthesized RNA molecule?
Well, there are several methods that are used to assess the quality of synthesized RNA to ensure that the RNA produced is of high purity, it's the correct size, and that it's functional. So all of these mean that the synthesis was a success. A few examples of ways to assess an RNA molecule are gel electrophoresis to check the size and integrity of the RNA, qPCR to verify that the RNA is of the correct size, and it's also another check for RNA integrity. And finally, mass spectrometry can be used to analyze molecular weight and functional analysis to confirm biological activity.
Well, this is science, so things definitely don't always go perfectly. So, what are some causes of in vitro transcription failure, and how do you troubleshoot or optimize those reactions?
Plasmid DNA is commonly used as a DNA template for IVT reactions. However, plasmid DNA is circular, so it needs to be linearized before it's used in the IVT reaction. And one common problem that scientists face is the preparation of the DNA template. So incomplete linearization of the DNA template can cause a big problem in IVT reactions. And this can be caused by use of damaged or contaminated restriction enzymes, because when a restriction enzyme is improperly stored, there's a decrease in efficiency, which tends to lead to incomplete or partial cuts in the DNA. Restriction enzyme star activity is another reason for incomplete linearization. Star activity refers to the enzyme cutting at non-specific sites. This often happens when there's too much restriction enzymes added to the reaction or when the reaction is incubated for too long. So, proper storage of restriction enzymes and preparation of the reaction master mix are both crucial for successful IVT reactions. The reaction buffer contains specific salt concentrations to provide an environment with the most compatible ionic strength to ensure optimal activity of the RNA polymerase and overall stability of the reaction components. DNA secondary structures can hinder the RNA polymerase's progress along the template. So, to prepare the DNA template for RNA polymerase activity, certain additives like DMSO and betaine can help relax the secondary structures in the DNA template to allow for more efficient transcription.
This is giving me water flashbacks to PCR back in the lab.
Me too.
Finally, when scaling up from research to production, what are the key considerations you have to keep in mind?
When scaling up, transitioning from research scale to production scale manufacturing of mRNA therapeutics, it really requires careful attention to scalability, consistency, and regulatory compliance. This includes things like process optimization, ensuring equipment suitability, and maintaining batch to batch consistency. This consistency is maintained through stringent quality control and adherence to good manufacturing processes, or GMP. The reliable sourcing of raw materials and an effective supply chain management system is also really essential, alongside comprehensive validation studies and thorough documentation to meet the regulatory requirements. Other factors like cost efficiency, environmental impact, and safety protocols also really need to be considered to ensure a viable and sustainable production process. Effective technology transfer and collaboration with external partners can further facilitate a really smooth scale up process.
Leah, thank you so much for joining us today and sharing your insights on RNA therapeutics and molecular cloning. It's been really fascinating to hear about the technology and expertise that goes into advancing this field.
Well, thank you for having me.
As always, thank you for tuning in to Teach Me in 10. We'll be back soon with some more quick, insightful science, so don't forget to like and subscribe so you never miss an episode.